students, greetings to all. In this module, we'll again continue with our ongoing chapter, Chapter 5, Principles of Inheritance and Variation. In today's video lecture, we'll be carrying out the discussion on multiple allelism. Now, the multiple allelism, that is referred to as the exception to the law of dominance that was proposed by Mendel. Now, multiple alleles, this term refers if there are more than two alleles for a particular trait on the same loci. When we talk about the diploid organisms, the organisms which have got the constitution 2N, they have got two alleles encoding a particular character. When we talk about the gametes, they have one allele which encodes for a particular trait or character. But in case of multiple allelism, two or more than two allelic forms encode a particular trait. Now, what are alleles? We know that they are different forms of the same gene which are present on the same loci or locus of a homologous chromosome. Now, the examples of multiple allelism are the coat color in rabbit. So, the example which we are going to talk about, that is coat color in rabbit and ABO blood grouping system. Now, when we talk about this multiple allelism, how does this multiple allelism occur in individuals? So, multiple allelism arises due to mutation in the gene. Now, mutations are the sudden, drastic, heritable or inheritable changes which get incorporated. Talking about the ABO blood grouping system, this system was proposed by Karl Landsteiner. And the genotypes, that is the blood groups A, B, AB and O, Felix Bernstein was the person to give an explanation about the four different types of blood group. So the person was Felix Bernstein and the system was proposed by Karl Landsteiner. Now, when we talk about the ABO blood grouping, we know that the various types of blood group are basically four. That is A, B, AB and O. The genotype which is encoding, so the genes which are controlling these various blood grouping patterns, that is AB, ABO are IA. So we'll be having the genotype that is IA, which is homozygous dominant. We'll have IA naught, that's homozygous, that is heterozygous dominant. For B, we'll write it out, the genotype as this is homozygous dominant. Again, we'll be having heterozygous variety. After that, we'll have the co-dominance exhibited by A and B. And finally, we'll be having I naught. So, basically, when we talk about this ABO blood grouping system, the genotypes, the allelic forms which control them, they are three. So, three alleles, they are going to carry out the expression of the four of the blood grouping patterns. Now, these three alleles which encode are IA, which is the dominant which encodes for blood group A, IB, again the other dominant which encodes for blood group B, and for blood group O, it's encoded by the recessive, so it can be written as capital I naught or it can be written as small I naught. Now, when we talk about the phenotype, so thus that means for the blood grouping pattern, we have got 1, 2, 3, 4 phenotype. So basically the phenotypes which are present, so 4 phenotypes which encode the blood group. And when I talk about the genotypes, so there are basically 1, 2, 3, 4, 5, 6. So there are basically 6 genotypes which encode for the blood grouping pattern. Now talking about the antigenic determinant. So the antigenic determinant which are present on the surface which help to determine the blood group. Like for example the RBCs, they've got the sugar receptors which help in the identification. So for blood group A, the antigenic determinant present on the surface of RBC will be A. For blood group it will be B, it will be B. For A and B, the antigenic determinants will be both A and B. And when we talk about the O blood group, there will be no antigenic determinants. So written it as N. Now when we talk about the antibodies, so the antibodies which are present in the blood group, so they can be for A blood group, it will produce the antibodies against B. B will produce antibodies against A. The AB blood group will produce none of the antibodies, and the O will have the antibodies for both A and B. So then in this case, the O blood group is referred to as universal donor because it does not have antigenic determinant, but the antibodies for both A and B. And whereas when we talk about AB, it is the universal acceptor because it does not have any antibodies present. Now when we talk about multiple allelism, thus the blood grouping pattern it is controlled by 3 allelic forms forming 14 phenotypic expression and encoded by 6 genotypes. So in this module, we had basically carried out the discussion of multiple allelism, taking up the concept of ABO blood grouping system. In the next module, we'll take the other example of multiple allelism, that is coat color in case of rabbit. Thank you.